All right, let's talk about practical chemical filters in this lecture. In this lecture, we will look at why physical chemical filters like Lipinski and Weber exist. These rules are not arbitrary, they reflect real biological barriers that limit which molecules can reach their targets. You will learn how to use these filters to define the chemical space your model should operate in, and why it is important to exclude molecules that violate basic physical constraints before training. Finally, we will discuss when real drugs break these rules, the so-called beyond rule of five space, and what that means for model reliability, generalization, and failure modes. So let's start with why rules matter before modeling. Um, we need to understand few things. Membranes limit how polar or how large a molecule can be and still cross into cells. Blood and gastrointestinal fluids place constraints on how hydrophobic a molecule can be before solubility and exposure matters. And molecular flexibility limits how reliably a molecule can maintain a stable conformation for binding. So together, these constraints define the realistic modeling domain for most small molecule ML tasks. They prevent you from training models on structures that will never be absorbed, transported, or distributed long before you ever compute descriptors or fit a model. Let's start with Lipinski's rule. Um, Lipinski's rule of five focuses on four simple molecular traits that strongly influence whether a compound can cross membranes by passive diffusion. These thresholds define a region of chemical space where passive diffusion is most likely to be viable. Molecules that are small enough, not overly polar, and not excessively lipophilic. Aspirin sits comfortably within this space. Its molecular weight, logP, and hydrogen bond counts all fall well inside the recommended limits. Um, that's why um, it passes every criteria, and that is the real purpose of Lipinski's rule of five, a fast, early developability filter to flag molecules with a uh, higher probability of oral absorption before investing in deeper experimental or computational modeling. And then let's see why Lipinski rules are not enough. Um, Lipinski's rule captures molecular size and coarse measures of polarity, but they miss two properties that strongly influence membrane permeability. So those are exposed polar surface area and molecular flexibility. A molecule can pass all four Lipinski criteria and still show poor oral absorption simply because its surface is polar, or because it has too many rotatable bonds leading to excessive conformational flexibility. These properties directly affect how efficiently a molecule can cross lipid membranes, which is why additional filters are needed beyond Lipinski's rule of five. Um, um, metoprolol and atenolol both satisfy Lipinski's rule of five, and at first glance, their basic descriptors look nearly identical. But when we examine properties, Lipinski does not explicitly capture, especially polar surface area. The difference becomes clear. Uh, metoprolol has a much lower TPSA. That means less exposed polarity, which allows it to cross lipid membranes efficiently. So the other molecule, atenolol, has a very high polar surface area and slightly greater conformational flexibility. That increased exposed polarity strongly limits passive membrane permeability, even though the molecule technically meets Lipinski's thresholds. So this is exactly why additional filters are necessary. Um, Weber's rule extends Lipinski by focusing on molecular geometry and surface exposure, not just composition. Empirically, a TPSA below about 140 square angstroms and no more than 10 rotatable bonds are strongly associated with good oral bioavailability. So these constraints limit exposed polar surface area and excessive conformational flexibility, both of which reduce passive membrane permeability. Weber's rule is especially useful for molecules uh, that already meet Lipinski's criteria but still fail permeability screens. Together, Lipinski and Weber define a much more realistic chemical space for orally administered small molecule drug design. Next, we will see beyond the rule of five. Lipinski and Weber rules describe most orally available small molecules, but um, many modern therapeutics intentionally fall outside those limits. Peptides and macrocyclic compounds often violate every classical threshold, yet a subset still achieves a meaningful oral exposure. The reason is, actually structural. So their 3D conformations can bury polar groups, form intramolecular hydrogen bonds and shield surface polarity. So reducing the exposed polar surface that actually governs membrane permeability. Beyond rule of five, chemistry marks the point where simple 2D descriptors stop being predictive and 3D conformation and molecular dynamics begin to dominate real world behavior. Next, we will see cyclosporine A versus cyclosporine E, and then you will understand what this is about. Okay, so cyclosporine A and E highlight the limits of rule-based filters. Both are large cyclic peptides, and both fail every Lipinski rule. Their molecular weights, hydrogen bond counts, and formal polarity all sit deep in beyond rule of five space. If you look only at traditional descriptors, there is no obvious reason why one should be orally bioavailable and the other should not. This is the key limitation of 2D filters. They describe composition, but not how that composition behaves in 3D space. I'll just show cyclosporin A and E structures, and then you can actually notice a very small difference, and that's what uh, actually matters. Um, the explanation only becomes clear when you look at the molecules in 3D space. Cyclosporin A contains a single N-methyl group that removes one hydrogen bond donor and stabilizes a cis-amide geometry. That small change allows the macrocycle to fold into a compact conformation, forming intramolecular hydrogen bonds that bury polar groups away from solvent. Whereas cyclosporin E lacks this N-methylation. It exposes an additional NH group, cannot stabilize the same fold, and remains more extended with polar surface exposed. So this is the essence of beyond rule of five behavior. Permeability is governed not by size or polarity, but by whether a molecule can dynamically shield its own polarity through 3D structure. So Lipinski and Weber are early screening tools. They convert medicinal chemistry intuition into fast computational filters for size, polarity, and flexibility. What we need to understand is that they are heuristics, not rigid rules, but they help you avoid searching chemical space that biology is unlikely to absorb. So the key takeaway from this lecture is use these filters to narrow the space before applying more expensive ML workflows. Thank you.